Hi, my name is Nicholas Maringo, and today we're going to go over super resolution single particle tracking, which will be represented by SPT throughout this whole movie. The advancements in optical microscopy to become super resolved were awarded the Nobel Prize in Chemistry in the year 2014. These were awarded for the work by Dr. Betzik, Dr. Hell, and Dr. Morner for their work in super resolution microscopy. So how small are the particles that we want to track, and how do we go about seeing them? Well, we use an optical microscope that is home-built into a custom setting, but we'll go over that later. To give you a sense of how small the particles are, a human is on the meter scale, cells are 10 to the negative fifth meters, cellular components are on the micron scale. Just below that is the size of viruses, and then lipids and proteins are on the nanoscale, and atoms are on the angstrom scale. The traditional light microscope can measure in these ranges, but in super resolution microscopy we want to measure in this region here at the nanoscale. Now let's go over how the intrinsic wave-like nature of light causes there to be diffraction in an optical microscope setup, and how that causes there to be an inherent resolution limit to which we can resolve things located close to each other. If you have a point source emitting light that is being collected into an objective lens and then passed through what is known as a tube lens in an optical microscope, the diffraction pattern of light that you get on your detector is known as the airy disk pattern. In 1873, Ernst Abbe described the ability to resolve two of these point sources located next to each other to be the wavelength of light you're probing with divided by 2 times the numerical aperture of your objective. This dictates the lateral resolution you can resolve in an optical microscope, and is usually around 200 nanometers. In order to break this inherent diffraction limit of light, most groups utilize a home-built wide-field microscope in which an objective collects your light, passes it through a dichroic mirror, and projects the fluorescent signal onto an EMCCD camera that is normally 512 by 512 pixels. You use, for example, a green laser that excites your fluorophores that are on the particles you're tracking above your objective. The fluorescent signal is allowed to pass through the dichroic mirror and is projected onto the EMCCD camera. This signal shows up on your EMCCD camera, and then you can collect movies at a 33 Hz rate in stacks of 1,000 frames. As mentioned, most of the particles you'll be tracking will be labeled with fluorescent dye, so let's go over that briefly. You have your incoming laser light represented by H nu, which excites a ground state electron to an excited state. Then the emission of a photon from that excited state gives you your fluorescent signal. Now that we've covered the fundamentals in optical microscopy and the setup, let's go over the steps to single particle tracking algorithms that you apply in the post-data acquisition. First, you want to increase the signal-to-noise ratio in each frame. Then, you want to localize each particle below the diffraction limit of light, and then construct your trajectories. In the first step of our tracking algorithm, we want to increase the signal-to-noise ratio in each frame. This is proportional to 1 over the square root of the number of photons emitted by your emitter. It's roughly 5 to 8 after acquisition, depending on your system. And the signal-to-noise ratio is very important and dictates the localization capabilities to break the diffraction limit of light in order to get super resolution localization. In this step, we're trying to eliminate some of the intrinsic types of noise that are present on an EMCCD camera, in particular shot noise, dark noise, and read noise. We do this regionally within one frame on a 50 by 50 pixel region of the 512 by 512 and apply a matrix of 1s to that region, iteratively increasing the signal noise 2 to 3 times. In the second step of our tracking algorithm, we want to localize the particles below the diffraction limit of light. Here in this frame, we can clearly see there are 7 particles in our frame of view, and the optical response at each particle is an airy disk. But if we look at the 3D photon distribution at that point, you get the point spread function. We can fit that point spread function to a 2D Gaussian, which gives us a lateral resolution of roughly 20 to 30 nanometers. And remember, this is dependent on the signal-to-noise ratio. In the last step of our tracking algorithm, we want to construct trajectories from these localizations. We use a nearest neighbor approximation in order to achieve these linkages between frame to frame. It's a visual representation of how these get linked from frame to frame. We minimize these false linkages by using a low probe concentration or changing the viscosity of the media we're probing in to slow down diffusion, or by post-analysis methods such as frame shuffling. So here, our final single particle tracking results can give us a high spatial and temporal picture of, say, proteins diffusing within the complex environment of a cell. And so this information is given to us in real time and is a high throughput method when done on a wide field microscope where we're collecting multiple trajectories at once inside of these complex environments. Lastly, let's go over the pros and cons of single particle tracking. The pros are that it eliminates ensemble averaging in systems when you use other experimental methods. Another big one is that you can perform these measurements in situ, which is huge in these complex environments, and also it provides high spatial and temporal information of these emitters. Lastly, the fluorescent labels can alter particles' intrinsic behavior, and with these fluorescent labels you always have to take into account that there's photophysical effects such as bleaching and quenching that can cause tracking to be challenging. And lastly, you use low emitter densities that don't always represent physiological concentrations.